The plasmid isolation from a culture of E. coli cells. Plasmid isolation is a common procedure in molecular biology used to extract plasmid DNA from bacterial cells. Here for this experiment, we have used the Escherichia coli bacterial cells, which are shown in a tube at the top. Inside each cell, there are circular plasmid DNAs represented in red-blue circles, and a larger tangled chromosomal DNA shown as a red tangle. The top right panel here shows the intact, double-stranded structures of chromosomal DNA red and plasmid DNA red-blue circle before treatment. The chromosomal DNA is a long, linear structure, while the plasmid DNA is a smaller, circular structure. E. coli cells after treating with lysis solution. The lysis solution has been added to disrupt the E. coli cell walls and membranes. This releases the cellular contents, including the plasmid DNA small red-blue circles and chromosomal DNA red tangled lines, into the solution. The dashed green outline around the cell indicates the breakdown of the cell membrane, with DNA now freely present in the solution. This stage shows the result after cell lysis and partial separation. The chromosomal DNA has broken down into fragmented, denatured pieces red dashed lines, while the plasmid DNA remains intact in its circular double-stranded form. This difference in stability and structure between the chromosomal and plasmid DNA allows for the selective separation and isolation of plasmid DNA in further steps. This bottom right panel illustrates the denaturation process. Under alkaline conditions, the chromosomal DNA denatures and partially unravels, becoming single-stranded and more susceptible to shearing. The plasmid DNA, however, remains more stable, retaining its circular form. Addition of neutralization solution. After the lysis step, where the cell membranes were disrupted and DNA was released, this image shows the result of adding a neutralization solution. The neutralization solution restores the pH of the mixture, allowing the plasmid DNA to return to its double-stranded form. The plasmid DNA remains stable as circular double-stranded molecules red-blue circles. Meanwhile, the larger chromosomal DNA, which had been denatured in the lysis step, cannot renature properly and instead forms insoluble fragments. These fragments, along with other cellular debris, begin to precipitate out of the solution. The side panel shows the state of the DNA molecules after neutralization. The chromosomal DNA red squiggly fragments is now broken into pieces, while the plasmid DNA red-blue circle remains intact and circular. Now the final part of this experiment is centrifugation. During centrifugation, the precipitated chromosomal DNA, proteins, and cellular debris form a pellet at the bottom of the tube.